I think this is a great segue into the next session, which is really about defining refractory patients in a more specific manner using IL18. And I will pass it on to Dan Lovell, who has kindly agreed to moderate our next session.
Thank you, Rashimi. So this next session moves forward with the concept about IL18 driven diseases, and we'll start the meeting, this part of the meeting, with discussion between Scott Canna and Fabrizio Di Benedetti, both of whom have been central researchers in this area of IL18 driven diseases. And then the next speaker will be Ed Behrens, who will give an example of a patient who had refractory systemic JIA who responded to IL18 blockade. And then we'll conclude the session with Chris Leptak, who has vast experience in regulatory aspects of using biomarkers for rare diseases. And so I'll turn the meeting over to Scott and Fabrizio. And I apologize in advance, I may have to leave before the end of the session, and Scott will serve as the moderator after 10 o'clock. Scott?
All right, can everybody hear me okay?
Yes.
Super, thank you. So again, thanks to Rashmi and all of the other patients who are helping drive this conference, because I do think that this is really sort of spearheading a lot of innovative things that are happening in peds rheumatology, especially from the clinical trial space. And there's certainly a staggering amount of unmet needs. So yeah, we initially called this session, Is It Time? But a trusted advisor said we should just say it is time for IL-18 driven clinical trials in autoinflammatory disease. So I'm going to be presenting, and Fabrizio is going to break in with color commentary at his leisure. So I'm also going to title this, If George Only Knew. So this is George Still, who coined Still's disease circa 1896. And so, this last session that we've listened to has been a lot of bandying about which anachronistic set of criteria that lump together diseases that we all think go together, but this is all gestalt and it's a garbage basket set of diagnostic criteria, no matter which ones you use. Why swap one anachronism for a slightly better one? And I think that we'll get to IL-18 in a second, but the idea of using a biomarker to unify sometimes disparate diseases is not unique to rheumatology. We've probably got the idea from oncology. And so oncology has been successful in a number of these, what they're called basket design trials, where you take multiple different types of cancer and you give a targeted intervention based on a biomarker that's common to all of these different types of cancer. And then you have a similar set of inclusion and a similar set of outcomes. And in this particular case, there's not even randomization. There's just looking at sort of baseline effects. And so, some of the key considerations when you're thinking about a basket design trial are, number one, biologic plausibility, the accuracy of the biomarker that you're going to be testing, the prevalence of the biomarker that you're going to be testing within the population you're interested in, how big the sample sizes you're going to need to be, and then whether your study designs are going to need randomization. So we'll touch on some of these, briefly. But, there's a number of examples of basket design trials ongoing or finished within oncology. So there's the NCI Match and NCI Impact. These are basically trials that are using either the genetics or the biomarkers of a tumor to determine what the treatment's going to be. I think there's someone who maybe could mute a little bit. And then there's been 3 drugs that have been approved for use based on cancer biomarkers, pembrolizumab and these. I'm not going to bore you with the names, but the point being that this is not entirely new ground for the FDA. The FDA has already done this in the oncologic space, but understanding that these are sort of numbers of patients probably larger than what we'd be able to enroll for our patients. So I thought I would just remind folks about the biology of IL-18 real quick. IL-18 largely comes from myeloid cells like macrophages, but also from the epithelium of our gut, our skin, or even in our lungs. it's made in sort of this pro form where it's inactive, and then it usually requires the inflammasome to kind of get activated and then get out of cells. But it doesn't just live outside the cells in an active phase. It's actually quite potently buffered by a protein called IL18 binding protein that binds really, really tightly and prevents IL18 from signaling. But in many of the patients that we're talking about, we know that they have just massive amounts of IL18. And we know that that's enough IL18 to sort of supersede their production of binding protein and result in systemic and measurable free IL18 that's now free to sort of signal on the receptors. And so what does IL18 do when it signals on its receptors? It mostly acts to augment the effects of other cytokines, usually JAK-STAT cytokines like IL-12, and it amplifies responses mostly in T cells or NK cells, but we're learning more about effects of IL-18 on other cells like ILC2s and neutrophils. And, you know, in those cells where it's been studied, it's basically an inflammatory cytokine. In cytotoxic cells, it helps cytotoxicity. And in cells that are making cytokines, it can massively augment their cytokine production, particularly of interferon gamma. So let's look at the IL-18 in the blood of these patients. And I have arbitrarily, this is my orange dotted line, at roughly 20,000 picograms per ml. And so, you know, this is a paper from way back in 2010. Healthy controls run here. I've summarized what we see in a lot of different diseases, as there's some diseases, like this is actually Kawasaki disease patients. There's other cancers and infections where IL-18 is by no means normal, but it is not to the levels that we see in what I'll call IL-18opathies, or what we've been calling systemic GIA and Still's disease, or at least a subset of those patients, where the levels are massively elevated. Again, this is a log scale. So what are these quote unquote IL-18opathies? So I'll start.
Can I ask you a question?
Please.
Can you go back to the previous slide? Why do you think that is I in a subset? If you look at this data, and if I look at your papers, I think you are the one who have published more on IL-18 together with Alexi. It really doesn't look as a subset. It's homogeneously increased in all patients we call SJIA.
I think that there is an experience of SJIA patients, and I think you know, one of our parents presented initially that she has a child with CNS involvement and arthritis whose IL-18 has never been particularly elevated. And so, you know, there are people who fit within this anachronistic clinical definition of systemic GIA whose IL-18s are not high, and they're probably, you know, a systemic presentation of someone who belongs more with polyarticular GIA, perhaps.
Would you consider that the exception of the rule?
You know, I'm so biased in the patients that I see that, you know, I'm like a bloodhound for IL-18. So I would say it's the majority of the patients we see present with IL-18 levels that are above this threshold, but it's certainly not all.
Scott, I wanted to ask a quick question. This kind of the complex between the IL-18 and the IL-18 binding proteins seems to have a very long half-life, and it's sort of very different from most of the other cytokines. Do you think this complex has some kind of a biologic function, and that there is a reason it stays in the body longer than what we see with other cytokines?
You know, I don't want to go off the rails on this. Yes, I've thought about this. I've talked with Aaron Ring, who is the person who is trying to weaponize IL-18 against cancer. The KD, the dissociation constant of IL-18 binding protein for IL-18, is at least as good as most monoclonal antibodies that have been affinity matured. And so what it would take to dissociate 18 from binding protein is, you know, pretty profound. So I think that remains a hypothesis. It also remains a hypothesis for things like anti-GM-CSF antibodies, where most of us have detectable levels. But, you know, whether there are circumstances in which binding protein lets go of 18, nobody's quite substantiated that yet. All right, so what are we talking about here? These are the diseases we've already discussed, and I'll call them syndromic. You know, so there's systemic GIA, which is rare, but not ultra rare. And there's certainly this IL-18 high, whether it's a subset or a majority. And these are the patients that have MAS much, much more than arthritis, and in whom I think we now have, some consensus that the arthritis is a part of, but not a necessary part of that phenotype. And I think, my recollection is that in the modern era, about half, maybe more, of the patients that get diagnosed with systemic JIA have a monophasic course with current standard of care. And so, yes, there is unmet need, but as Fabrizio pointed out, many of these patients are being treated successfully with existing treatments. There's MAS, which is a rare temporal complication of systemic JIA. I like to point out that MAS is not like a light switch. It's not as though patients either have it or they don't. I think many, if not most, systemic JIA patients have features of MAS that, have to get to a certain threshold before they would meet criteria for what we would call fulminant MAS. And again, in most of our MAS patients, we estimate, but we don't have good numbers, that maybe half of these patients will get into remission, often with aggressive treatment, but with existing approved treatments. Then there's, of course, the refractory systemic JIA population, which is probably enriched for this IL-18 high subset and has high unmet medical need. And this, of course, includes systemic JIA lung disease, which has been discussed at great length. But there's a few important monogenic causes of chronic high IL-18 that I think are relevant because they put IL-18 upstream of pathology. And maybe the easiest is this NLRC4 related disease. Again, these are ultra rare. We're talking about patient numbers, probably globally less than 50. There is one case report of IL-18 blockade being efficacious in NLRC4. And there are two ongoing clinical trials, one of which includes only quote unquote elevated total IL-18 as an inclusion. And I won't sort of belabor, but there are other monogenic autoinflammatory diseases associated with chronic high IL-18 and autoinflammatory phenotypes. More recently, we published that a slightly different phenotype that's not a systemic cytokine storm MAS disease, but a skin arthritis disease also has sort of chronic high and unopposed IL-18 and is also quite unmet medical need. There really is no standard of care for PAPA. So how do we test for total IL-18? Well, when we measure total IL-18, most of us send it off to Cincinnati. I believe a couple other labs like Mayo are also running it. It's easy. If I can do it in my lab, it can't be that hard. And we've been doing it for a while. It has a very broad dynamic range, so it really helps you divide out who's normal, who's a little bit high, and who's in the sort of IL-18opathy range. It's quite reliable and stable to freeze thaw and shipping. It's associated obviously with a biological activity that we can target. And we know that because there are phase 2 and 3 programs targeting this. There is not an FDA approved test. It's not like you can just order this test for your hospital. It's being performed in CLIA-CAP certified labs, but there's not an approved test. And if the FDA felt like in order to do these kinds of trials, you needed to develop a companion diagnostic, that's maybe something for sort of venture philanthropy, and we can discuss that. As I mentioned, when we measure it, we're measuring mostly this complex and not necessarily bioactive IL-18. And so the cutoffs that we might use, we certainly have data to inform them, but they're somewhat arbitrary. Now, this is a meta-analysis of IL-18 in systemic JIA and MAS. And so I would say in our diseases, if there's enough studies for a meta-analysis, this is well studied. So here's my arbitrary 20,000 cutoff point. Here are healthy controls, a whole bunch of other inflammatory diseases, MAS and systemic JIA, and even HLH, or the quintessential cytokine storm disorder. And when you set your 20,000 cutoff, you pretty much eliminate everybody that isn't an IL-18opathy. Now, in systemic JIA and MAS, IL-18 is not always and uniformly elevated. This is an old case report from 2010 where, IL-18 levels, almost never normalize in this patient, but certainly go up really high, in some cases close to 1 million during MAS episodes. This is LDH to sort of as a surrogate for MAS episodes. And here's a paper that we published on a handful of patients most of them refractory systemic JIA, 1 or 2 NLRC4, who we followed over the course of years, whose IL-18s never got close to normalizing, were always above 20,000. So there is a point at which it seems fairly clear that total IL-18 above this arbitrary threshold of 20,000 basically is associated almost uniformly with detectable free IL-18. And so this is again from our 2018 study. And, you know, all of these sort of above normal and in some cases fairly high IL-18 levels, when you measure free IL-18, sort of go away. And I'll point out that the instances in which we see really high IL-18 outside of systemic JIA, MAS, or NLRC4 are, you know, this is malignancy-associated HLH. So these are patients that would be excluded by any exclusion criteria from any study that we're discussing. So the idea of a basket trial in autoinflammatory diseases is not unknown. You know, it's already been done by Fabrizio's group in colchicine-resistant or refractory FMF, hyper-IgD or mevalonate kinase deficiency in TRAPS, where they used Ilaris. This was a placebo-controlled, I believe this was sort of a divided basket where this was basically 3 different trials all sort of rolled into one. And their inclusion and exclusion were basically the genetics and then a flare defined by CRP and a physician global. So this is not exactly the most you know, disease-specific inclusion criteria, apart from the fact that they required a gene. And we know that there can be a lot of heterogeneity in genetic diseases. So these are the ongoing IL-18 blocking trials. One in Still's disease used a recombinant form of IL-18 binding protein. This was a phase 2 study that was open label with a dose escalation, and it was mostly a safety study. There's another study ongoing of another IL-18 monoclonal that is basically the same type of study. Importantly, neither of these studies require or even look at IL-18 at inclusion. And I think that this is something that has, at least for the published study, hampered the efficacy of what they saw, because a good subset of their patients never had very high IL-18. So why would it work? And then these are the 2 studies in monogenic diseases. One, again, using recombinant binding protein, and another one that's just open using an IL-18 sort of bispecific antibody. These are open labels with randomized withdrawals. And so the primary endpoint is the time to flare in the randomized withdrawal phase. And again, the inclusion are wildly nonspecific except for the genetics, CRP or ferritin elevation. And in the former, they used a modification of the ADI, which I'll show. And in the latter, just the physician global. And again, for this study, they just said the patients had to have elevated IL-18, but no threshold was described. This is the autoinflammatory disease activity index. And as you can see, it's a little bit like the autoinflammatory SLEDAI. It includes a whole bunch of different clinical manifestations. The modification being used in the first NLRC4-XIP study adds some of the clinical features and some of the lab abnormalities that indicate organ involvement in some of those diseases. So one option for an IL-18 driven trial is just to add IL-18. Pick your IL-18opathy, whether it's refractory Stills or NLRC4, and you just say you have to have IL-18 above a certain threshold, and now you might sort of biologically subset a heterogeneous group into one that may be more likely to respond to your targeted treatment. You know, the problem here is, again, you're dealing with ultra rare diseases, so enrollment might be a real challenge. And then you're not studying all the diseases you're not studying. So, you know, you might get an indication for systemic JIA, but not for, you know, any of the other diseases that you're interested in studying. So one option to do that is to do like was done in the Ilaris study that Fabrizio did, where you basically lump all these different diseases together, have different inclusion criteria for each subset, and basically run what looks like multiple parallel clinical trials for each disease. But they're just done at the same time with a very similar clinical trial structure. And so, you know, the IL-18 cutoff that I arbitrarily threw together, but I think the data supports, is you could say something like 2 IL-18 levels above 20,000 separated by at least 2 weeks, or a very high level above 50,000 once, in addition to whatever clinical criteria you're going to use, CRP, ferritin, physician global, or modification of the ADI. And obviously, with the exclusion of intercurrent infection or malignancy. This is an autoinflammatory disease. You cannot define it as autoinflammatory unless you've excluded a major contribution of infection or malignancy. So the pros here are you cover multiple indications. You don't get sort of elevated by one sort of dominant indication that brings up other diseases that may not have responded, nor do you lose the effect if there's one disease that doesn't respond and it sort of tanks the statistics of all the others, balloon or anchor effects, I'm calling them. And you can tailor your inclusion to the nuances of these diseases. Again, in order to get any kind of statistics for individual diseases, you're going to have really challenging enrollment. And not every disease is going to be studied, and that would remain a neglected disease. And so why not just define an IL-18 associated autoinflammatory disease where you say you have to have an IL-18 cutoff, we know which diseases meet that, and then you define an autoinflammatory disease like we have for all these other trials with CRP or ferritin, and then something even as simple as the physician global or the MADI with, of course, the autoinflammatory exclusion criteria. This is a design that sort of meets the greatest unmet need. It's the maximally biologically informative, and you could actually potentially enroll enough patients to get some statistical power. But of course, you do have a risk of there being either some subsets that do or don't respond that will drive or tank your statistics. And it's possible that some of your outcomes may be too diverse for the measures that we have. But I would say that existing trials are using outcomes that are already quite nonspecific. So how do we move this forward? Well, first, I think we need some appetite, both from industry and from regulators, to actually define diseases biologically. And we have the additional challenge that not only is biomarker-driven trials a novel concept, but we're doing this in a really, an orphan space. But that's really where the unmet need is for these patients. Perhaps we need better population estimates of incidence prevalence of each of these subsets. And then, that's something that could potentially be solved by the natural history data that we'll be studying later. But, there is a way of sort of having a more formal meeting about this critical path innovation meeting where you bring together physicians, patients, industry partners in front of the FDA and say, how do we get this done? And I'm hopeful that we can make that happen. So I know I've gone way over. These are all the people that are in our community, and we love you all. And I'll stop talking.
Okay, next speaker is Ed Behrens. He'll talk about an example of a case.
And I share my video, I guess, right? Okay.
Correct.
Okay, desktop 1. Sorry, now I'm getting something's different with Zoom. Why am I not able to Zoom to share my screen?
Do you see the share screen button, Ed?
Yeah, no, it's I'm getting a security message that I need to change my preferences. I'm not sure why I am getting that because it looks like it is set up to share. Apps. Zoom is checked off. I've not had this problem before because obviously I use Zoom all the time. This is going to be a fail, huh?
Do you have a PowerPoint presentation you can send by email?
Yeah.
Would that be okay?
Yeah, I sent it. Who do I send it to? I sent it to Rashmi yesterday.
I have it, and I can just start it up in just a sec. I'm just yeah.
Okay, I'm sorry, guys.
No worries. Okay. So let me just share my screen, and it should be up there in just a moment. Okay. Yeah.
Great, thank you. Yeah, so I'm going to talk about, so we've been talking about trial design, which includes lots and lots of patients. We're just going to talk about an N of 1 here. So, obviously all the grains of salt that need to be taken with talking about a single case, but I thought it was relevant to our discussion about maybe why it is worth trying to think about whether IL-18 should be our next target for systemic JIA. So thanks both to the physicians that helped put this together, as well as to the patient and family that, took a real chance in trying an unknown drug for their condition. So next slide, please. So, I'll try to go through stuff really quick just to try and get us on time. So, really brief case history. This actually is an easy one. As much as we've debated about classification criteria, this one checks all the boxes, right? So this was a child that met ILR criteria, had fever, had arthritis, had the rash, presented at an early age, was in the phenotype of having multiple episodes of MAS, and then around 7 to 8 years of age, developed lung disease. This included inflammatory clubbing, a decreased DLCO, cough, and a chest CT with extensive peribronchial inflammation. And also has the HLA-DRB1 risk allele that is associated with lung disease and systemic JIA. So this meets all the boxes. We don't necessarily have to question the case definition here, and I'll give you some more data to support that. Next slide, please. So this is a picture of the biopsy, just showing the endogenous lipoid pneumonia. You can see the cholesterol clefts obviously present in the lung tissue. You can also see extensive interstitial lung inflammation here as well. Next slide. Lots of foamy macrophages eating presumably the proteinaceous material that went along with this patient's PAP. Next slide. This was the BAL, which showed multiple globular deposits of protein consistent with PAP. This is just one example of one field from the BAL slide. So, I don't think we need to debate the case definition here. This child literally checks every box you can check for the systemic JIA lung disease that we're talking about. Next slide. So, like most of the patients in this situation, very refractory disease. So I'm abbreviating a really long history in like 4 bullet points here. Prior treatment with methotrexate, canakinumab, prednisone, after coming to see, and by the way, refractory arthritis to, even while on these medications, as well as the progression of lung disease. When they came to the CHOP clinic, we decided to just try to aggressively treat, knowing the sometimes dire prognosis of lung disease in this condition. We used a 4-month cycle of cyclophosphamide followed by azathioprine maintenance. And this really sort of stabilized things, both from a perspective of clinical inflammation or systemic inflammation, as well as the lung disease, for quite some time. I will point out that we did attempt a withdrawal of canakinumab while on the cyclophosphamide, which resulted in a significant reflare of systemic JIA symptoms. And so we really did need to restart the canakinumab to get control of disease. This is pursuant to this idea that canakinumab and other biologics may actually be a cause of the lung disease. In this case, withdrawal of the medicine actually made things worse. And then disappointingly, the benefit we got from the cyclophosphamide and azathioprine was short-lived, is probably too pessimistic, but didn't last longer than 12 to 18 months. There was a reoccurrence of shortness of breath and cough. And so we went on to other agents that I'm sure everyone here is familiar with: tacrolimus, tofacitinib, increases in prednisone. And all of this was basically unable to control the lung disease. There was a progression of both clinical symptoms as well as worsening of pulmonary function tests. And the pulmonologists, were basically getting very agitated that I needed to do something more to try and help the patient. Next slide. The biomarkers are all in line with all the things that you would expect. IL-18 in the serum was very high, usually greater than 100,000, but always quite high. Again, independent of maybe whatever the other systemic inflammatory markers were doing at the time. With the help of Scott Canna, we measured free IL-18 both in the lung and in the BAL fluid, and there was detectable free IL-18 in both. Again, consistent with a bioactive IL-18. The CXM9 was elevated. This did normalize with tofacitinib use, but did have an elevated CXL9 off that medication. And then, you know, his LDH was always high. I'm never quite sure what to do with this lab value, because it never correlated with anything clinically important, but it was certainly always high. Next slide. So given the situation and the fact that we had pretty good evidence for bioactive IL18 in this patient, both in the lung and in the blood, we decided to try an expanded access investigational trial of the MS-825 drug product in this patient. For those of you that don't know, this is a bispecific antibody, one arm that blocks IL-1 beta, the other arm that blocks IL-18. And we're now, I think, 8 months into therapy, and I have to actually count it up. And again, I'm going to just summarize a bunch of data really quickly. I apologize, I don't have sort of beautiful data slides, but I think for the sake of time, the summary is good enough. Lung symptoms have improved with cough going away, increased exercise tolerance. Sort of subjectively, when you ask the patient, there's a very clear overall improved sense of well-being. We were able to discontinue the tofacitinib 2 months into therapy with no change in the CXL9, so the CXL9 did not rebound off tofacitinib. We've been slowly working to wean the prednisone off. I think everyone will appreciate that, given the large burden of prednisone and the long duration of prednisone, it's not a medicine we can just like flip the switch on immediately. But I'm happy to say that the patient is on the lowest dose, sustained dose of prednisone that he's been on in 2.5 years. We are anticipating that the prednisone will be off, I think, in the next month, if I have my timing right, but hopefully soon. And so, you know, these sort of subjective things are all nice. Next slide.
Yeah, did you repeat the chest high resolution chest CT?
I'm getting to the I'm getting to the objective findings now, Alexi. The IL-18 went down to less than 500 after the first dose. And as I mentioned, no change in the CXL9 even after tofacitinib discontinuation. Now that lowering of IL-18 could be something to do with we've altered the IL-18 biology, or it could simply be that the antibody is in fact interfering with the IL-18 assay. But either way, the drug has hit its target, and that happened even after the first dose. Interestingly, as much as I poo-poo the LDH, I will now have to sort of eat crow and admit that his LDH has been steadily declining over time as you measure it. So it seems to be measuring something and doing something and improving. So for whatever it's worth, the drug actually, of all the other drugs that this child has been on, this drug is actually the first one to actually move that biomarker. His 6-minute walk distance has improved by 40 meters. His post-exercise pulse ox has improved by 3 to 7%. His PROMIS mobility scores and global physician assessment scores are also both with sustained improvements, which I'm quite happy with, because that's a marked change. Unfortunately, Alexi, there has not been a change in his lung CT or his DLCO. They've stabilized, which we're happy with, because they've been progressively getting worse, but they've not necessarily improved. And again, it's hard to know what is irreversible fibrosis and damage versus what is what actually might be reversible. Next slide, please. What does seem to be reversible is lung inflammation. So his pre-treatment BAL cell count was 181,000 cells per ml. Post-treatment, we were able to decrease to 30,000 cells per ml. And those proteinaceous globules are now completely gone. I was sort of joking with Scott Canna. You're actually looking at what the slide looks like on this blank slide. Like you don't see the protein globules anymore. It's relatively acellular. Next slide. Super preliminary, but I just tried to throw together some interesting stuff. We measured all the things, right? So I am cherry picking a subset of data here. And what I cherry picked for you were things that changed in the BAL that didn't seem to change so much in the blood, right? So these are sort of lung specific changes, if you will, that the drug sort of induced. Now, I'm going to remind you, what you're looking at here are percentages. If I gave you absolute values, because the absolute cell counts in the BAL went down so much, everything just tanks, right? So this is sort of asking the relative proportional changes of the types of cells that are in the BAL pre and post treatment. So the number of these sort of CD69 positive Treg cells, these are CD4 Tregs, sort of I think strikingly went up as a matter of percentages in the BAL post-therapy. I love memory CD8 T cells, and I could talk to Alexi for like 5 hours about the importance of tissue-resident memory CD8 T cells in liver inflammation and interferon gamma-driven liver inflammation. And I wonder if that's relevant in systemic JA. But if you look at them in the lung, they actually seem to have decreased and or switched phenotype away from being activated with becoming CD69 negative. In the myeloid compartment, I found it interesting that there was sort of an increase in CD32 positive or increase in the amount of CD32 and the amount of CD4, CD16 positive macrophages. Maybe they're doing a better job eating up the protein that doesn't belong there. And then sort of interestingly, again, as a measure of, are we getting at that 18 gamma axis, HLA-DR went down both in the blood and in the lung. Next slide. So to just quickly sum it up, N of 1 study in MS-AT5 and systemic SJIA-LD, subjective improvement in pulmonary and global symptoms, objective improvement in laboratory and functional measures, decrease in prednisone and non-prednisone drug burden. And I'll just sort of, I don't want to sort of have all the guns come out blazing at me, but I think it is important to point out that the excipient that's used in the MS-825 preparation is overall similar to the excipient used in all the other monoclonal antibodies that patients get. So with respect to this hypothesis that there is some magic thing that is common to a lot of drugs that is driving the lung physiology in systemic JA, at least in this N of 1, the same stuff was in this MS-825 that seems to be making this child better. So take that for what you will. And hopefully we're getting closer to being able to say the time is now for trials of IL18 blockers in systemic JA.
Thank you. Maybe in the interest of time, we should move on to Chris. Scott?
Yep. So we are fortunate. We're unfortunate to be going so over. I think we bit off more than we could chew this session, but we're fortunate to have Dr. Chris Leptak, who is currently at, and you'll forgive me if I forget the name of the consulting firm, Greenleaf Health, which is a consulting company, but he was with the FDA in their oncology branch for many years and was really one of the pioneers of some of these biomarker-driven designs. And so we're really hoping to get his experience and his reflections on what we've been discussing.
Excellent. Thank you, Scott. And thank you, Rashmi, for the invite. Certainly this space is very near and dear to my heart, not the least of which because I have a PhD in immunology and did cytokine regulation back in the day on a hepatic parasite. So certainly very thankful to speak to you today because of the time. And as I shared with Rashmi, even when she reached out, the Q&A is really the crux of this so that folks can get the questions that they have answered. So Coralie, I know you're helping with the slides. Honestly, I think if we just focus on slide 8, that will kind of set the stage for the Q&A. The rest of this is background material. Since Scott and some of the other presenters have already touched on this, I think this will kind of be a nice summary. So the first thing I want to convey is that certainly oncology gets a lot of the press around the use of biomarker-driven trials. Please don't think that that's the only space where that occurs, because it occurs throughout all the therapeutic areas, perhaps oncology a bit more frequently, but I don't think you should be discouraged around a biomarker-driven trial outside of the oncology space. And that's also true for the rare diseases as well. So this is something that the FDA clinical divisions are quite familiar with. And the different trial designs, even some of the ones that you proposed today, are things that they can certainly have conversations around. I also want to put in a pitch for the critical path innovation meeting that Scott had mentioned. This is an opportunity based on the participants in today's call. You have patients, patient groups, you have academics, and you also have the pharma community represented, that those critical path innovation meetings are an opportunity to have a conversation with the appropriate experts on the FDA side outside of the normal drug development IND meeting process. And so if you wanted to posit a potential IL-8 driven trial and what that might look like and the endpoints that the FDA might consider, I think that's a great first start for having those initial conversations. Certainly for the drug developers that have their therapies in play, they can follow their normal IND meeting process engagement. So a couple of the thoughts on this slide, some of the speakers have already hit on, and Rashmi had challenged me to say, well, how can biomarkers be incorporated as part of a clinical trial in the setting of a rare disease, and even in the setting of rare disease with a very heterogeneous presentation? And certainly by this morning's earlier comments from those of you that are the subject matter experts in the space, which I'm not, some of the challenge you're facing are not uncommon, unfortunately, around some of these disorders where patients present very differently and they have different experiences of their disease, and those experiences may change over time. They're not necessarily constant. And so I think a biomarker, introducing biomarkers as part of the clinical trial design would certainly be appropriate and something that the FDA would be open to. So the first is around enrichment strategies, and Scott and others had mentioned that this might be an opportunity. If you had an IL-8 as part of the inclusion criteria, that could be part of the diagnostic for your IL-8opathies, as Scott referred to it. Certainly establishing a cutoff for enrollment would be important. Scott has his 20,000 cutoff. Certainly as a community, you guys decide what that cutoff would be as an opportunity. There's also prognostic enrichment, where if in fact the IL-18 is correlating with disease severity, and because these are chronic diseases that have maybe a waxing and waning character, you could establish an IL-18 that correlates with the level of disease severity, and then enroll a subpopulation of patients that are more likely to have whatever your clinical event or outcome of interest is within the time frame of the clinical trial. And so that might be another opportunity. And then finally, there's predictive enrichment, depending on the mechanism of action of the therapeutic that's involved, whether it's an anti-IL-18 or some other mechanism. You may find that only a subpopulation of patients are expected to respond, and you could enroll those patients based on whatever biomarker background you want to consider. Certainly as drug development continues and you're going into phase 2, you want to show proof of concept. If you have an anti-IL-18, you want to demonstrate that you're actually hitting your target and reducing IL-18 levels as your proof of concept. So that's another use of the biomarker. Kind of analogous to that around dose. If you're going to be studying multiple dose cohorts and you have a target in mind of how much you want to reduce IL-18, you could follow IL-18 reduction to figure out which dose would be the appropriate one to potentially carry forward in your phase 3 program. If in fact these IL-18 opathies are IL-18 mediated, by definition, you would hope that ultimately you can see a treatment response by reducing IL-18, that you're going to see an improvement in your clinical outcomes, however those are going to be defined. And then finally, it may be possible in an accelerated approval context that IL-18 could be an endpoint, perhaps a co-primary. I don't think that the IL-18 reduction alone would get you an approval for accelerated, but it may be along with a clinical outcome as an opportunity to demonstrate that, in fact, the therapy works in that population of patients that were enrolled. And then in the post-marketing setting, you could do additional confirmatory evidence. So I think those are some of the thoughts that I had based on the knowledge that I have of your space, which is somewhat limited. But this is consistent with what other approaches are doing in the rare disease space that are focusing on a biomarker-driven design. So with that, I think we're going to have a panel discussion, and I'm certainly open to any questions people might have.
So while people are formulating their questions, I'm going to ask Dr. Nikolov, who I have in the chat put on the spot, for his reflections, thoughts about this. There's very little experience with biomarker-driven trials, especially in the orphan rheumatic space.
Yeah, thanks for putting me on the spot, Scott. And I'll try to take this one. I think actually Chris gave a very good framework around how to think about the use of biomarkers. And just for clarity, in this case, IL-18 would be considered a biomarker. I think ultimately the approach that we take, or the advice that we give to drug developers, is based on the data supporting a specific biomarker. So how much of the science is known about this biomarker, and how much the consensus there is on the utility of this biomarker for a specific disease. Just to give you an example, CRP and ferritins are biomarkers, and they have been successfully used to enrich patient populations for clinical trials. So if the intent of the biomarker would be to enrich for a patient population, I mean, we certainly have precedents for that. I think there are additional considerations of what additional data might be needed for this biomarker to be used, because I'm not sure that this is widely available or even commercially available, and whether this could or should be used as a companion diagnostic, for example.
So is that a requirement that that if you're going to use this in a trial, that it has to be an FDA-approved study or FDA-approved test?
So I think it depends on the context of use, but if the intent of the use is to select a patient population, to define a patient population for a specific intervention, again, for regulatory purposes, during the development, a biomarker may need additional information. And again, there are other parts of the agency that are involved in the validation or the assessment of these biomarkers as companion diagnostics.
If it's obtained in a clear certified lab, would it satisfy you, or it needs to be approved by the FDA?
I think I may need to reach out to our CDRH colleagues to address some of these questions. But I think the point is that the test should be readily available and interpretable by an average clinician who would be prescribed. Well, not average clinician, because in most of these cases, you know, subject matter experts really manage these patients. But it should be available in a way that a clinician should be able to define this patient as a patient who meets the criteria from the clinical study, and we know that the drug is expected to work in those patients. Now,
sorry, go ahead.
Well, Chris, by all means, you're probably more of an expert in that field than I am.
Around the sort of companion diagnostic question, Scott, in all likelihood, if you're going to have the drug approved for an IL-8 as part of the indication statement, the CLIA approved test likely won't cut mustard. I can't speak to that specifically, but just based on what I've seen, and they may want additional evidence to show that the CLIA labs, which all have their own sort of assay that they've developed, in fact, have similar performance characteristics, because we know that the variability across those is quite large. And so if you think that you're going to have an IL-18 driven trial design, it would behoove you to at least think of a diagnostic partner that could engage with CDRH through the pre-sub process to at least see if there's something that can become commercially available and vetted through the FDA, because that would it would certainly prevent any unwanted surprises further down the road.
Yeah, thanks, Chris. And again, I think I don't want to go into the details about the assay or the test itself. To me, it's more of a more high-level discussion of your proposal. So if you want to use a biomarker to enrich patient population for clinical studies, that's one question. Now, what I heard is also that you propose that you define a disease called IL-18opathies, if you want to call it this, sort of lump all different disease entities or conditions based on a biomarker. And I think this may need further consideration. Again, you use the example of site agnostic cancer therapies. And I think for cancers, we can go into this discussion, later on, but I think the case of cancer might be different than the case of complex immune-mediated diseases, where it's not only IL-18, it's multiple other components of the immune system involved that could be important or relevant or driving the disease.
Yeah, I would challenge that.
Again, I can only share kind of high-level considerations for you to think about, not necessarily, that we need to debate this here, because you guys are the subject matter experts. But I think you also need to consider whether you would be increasing the heterogeneity of this patient population that ultimately can make it more difficult to demonstrate efficacy. Because a decrease in IL-18 levels, with a certain intervention is probably not going to be sufficient to demonstrate efficacy of an intervention. So if the intent is to show a clinical benefit, I think you need to consider the patient population and the homogeneity on the clinical manifestations that you want to show benefit on.
So just to bounce something right back to you, we wouldn't actually be using dropping of IL-18 as an outcome measure, because in some patients, especially the genetic ones, it may never come down even if they're clinically well. So the outcomes would be the clinical outcomes that we're using in things like systemic GIA trials. I think that the trade-off that we're considering here is, you know, trial design versus enrollment. Like if we're going to restrict to really small orphan diseases, whether they be genetic or SJIA-LD, you get into a situation where it becomes, you know, as Randy mentioned earlier, you know, open label, non-placebo studies, which our experience has been that that's not been something the FDA would consider as part of a registration study. So, you know, is the FDA tied to having some placebo-controlled portion of a trial as necessary for approval?
So again, these are loaded questions that you're asking, and I'll try to go back to the basics that I think it's not for FDA to define a disease entity. And if you think that the disease entity should be IL-18, you know, opathies, it's for the scientific community and the broader community to define these as disease entities. Now, for a proof of concept, whether, you know, targeting this pathway might be relevant to multiple diseases, certainly, you know, studies could be done. Whether those studies would be sufficient to support regulatory decisions, approval, and so on, that's kind of a separate question. Ultimately, I think we would expect to have reliable, controlled efficacy assessment data for a defined disease entity to be able to make a reasonable benefit risk assessment for that disease. And that doesn't need to be hundreds of patients, especially for effective treatments. But back to your example also about canakinumab and the basket trial. In that trial, each disease entity was a standalone entity where each of these sub-studies demonstrated efficacy for each of these entities. So we didn't treat it as a basket of periodic fever syndromes. We treated them as, you know, HIDS or, you know, hyper IgD syndrome and TRAPS and FMF. So I think you're asking too many questions that are, you know, very challenging for us to address at this point. But I think what we do is based on the science and what you guys are generating in terms of support of use of, you know, biomarkers to select patient populations, endpoints that can be reliably assessing a clinical benefit. So I'll pause here, and I know that Fabrizio wanted to comment.
Thank you, Nikolai. I think what you're just mentioning is very important. You're mentioning on the one hand that we may be using a biomarker to select, and I'll stress the plural, populations of patients that are clinically different, but they have to be homogeneous, which, and you mentioned the basket trials in which each disease, the canakinumab basket trial, in which each disease entity was considered as a separate entity. So I think that even if we are in the setting of a rare disease, which we should think about basket. Scott, I'm looking at you. We should think about basket, either sequentials or contemporary basket, based on, you know, interleukin 18. I think we cannot lump together all IL-18opathies. This would be my view. We should try to define disease entities that we think are relatively homogeneous, particularly because of the outcomes, which is something that Nikolai was alluding to, I think. The outcomes are going to, the way we measure the outcome, even if we use PGA and CRP, which is fine, are going to be probably different. In this, I love the slides of your monogenic and syndromic IL-18opathies. But the outcomes are going to be different, Scott.
Yeah, I totally agree. I'll point to the N numbers that you got in that sort of divided basket trial. You know, you had 46 TRAPS patients, you had 60-some CAPS patients.
We played very safe. We played it very safe. But if you look at the numbers, 15 or 20 patients per disease would have been sufficient.
Yeah.
So we didn't know what we were doing. Now we know more, I think.
But I mean, you know, we know how many people on this call know how hard it was to recruit 10 patients for a study that included both NLRC4 and XIAP.
I know.
And so
I know.
You know,
we know.
The basket design is.
I know.
So I think that's maybe the challenge that we're dealing with. So I know that Chris, you have a hard stop at 10:15 that we just passed. Any last thoughts before we lose you? You're muted, sir.
The joys of the virtual presentation. No, again, I don't think you should be discouraged. Certainly amongst yourselves, you have differing opinions, just like on the FDA side of the fence, you'll have differing opinions. But the important thing is to have those conversations and to see what might work. And as part of a phase 1 study, that may not be how your phase 3 is going to look. Because you're going to learn from your phase 1 and 2 of what your population is going to be the best to target, depending on the mechanism of action of the product. But, you know, just to stress Nikolai's point, in the one I made earlier, you know, reduction of IL18 alone is not going to get you out the door. You're going to have to show that clinical outcomes of interest. And that's where the basket approach, as Fabrizio has pointed out, gets to be a bit problematic, because those patients are going to have different outcomes that are going to be relevant based on their presentation of the disease.
But Chris, just to intervene, we won't be able to do a phase 1, 2, and 3 study here.
Right.
We'll be able to do one study and one only study. Okay, so it's a bit more complicated because we have to get it right.
And I'll point out,
we have one option only.
We're talking about the outcome measures here. What are the outcome measures that you all have done? It's PGA, it's CRP and ferritin, right? These are not specific outcome measures to systemic GIA or to CAPS. You know, you didn't even use the ADI for the basket trial in autoinflammatory disease. It was not
validated at that time.
So Rashmi's right. We should wrap up, but I think that I think that we could have a heated FDA. What's the acronym again? The CPIMS study.
We need the critical meeting. Yes.
The one side of this we haven't heard from are the people sort of footing the bill for all this, is the industry side. And unfortunately, there's been some personal issues preventing some of those folks from joining us, but I'm sure they're listening and very much part of the conversation. So Rashmi,
anyone from industry want to comment? Not. But we can continue this, and in today's working session is actually devoted to this, so we can continue this after the stem cell transplantation session, which is right after the break. Not that we have much of a break, but I want to Scott, you want to just wrap up so that I can.
Sure. Is it the same Zoom link, or is there a different?
No, it's a different Zoom link. And if you are logged in via EventAir, then you need to come back in to EventAir.
So the next session. So thanks everyone. This has obviously been very spirited and heated, and I think there's interesting biology and interesting clinical work to be done. The next session is actually about stem cell transplant in refractory systemic GIA, especially in lung disease. Something I am moderating, but far from expert. But we have a really pretty amazing panel of speakers, and I encourage folks to join.
Yeah, and before I wrap up, thank you so much, Nikolai, for being here, and everyone else from FDA, etc. It's really helpful to have you here and to give us some guidance. So thanks, and we'll be back soon.